So this is going to be a review on Unit 2, okay, which is on cell biology. Your corresponding textbook chapter is going to be from Chapter 4, A Tour of the Cell. Okay. Before we move on to each component of the cell and the functions of each of these components, first, you have to learn the technique that we use in order to study cell biology. And this technique is called cell fractionation. A-C-T-I-O-N-A-T-I-O-N. So cell fractionation, right? Fraction means it breaks it into different parts. And cell fractionation is a technique used to study cell structure and function. Study cell structure and function, and this technique is often being mentioned in the test. So here the idea is they are going to separate the cell's components into different layers. The idea is first you have a bunch of the cells culture, right? You're going to break apart the cell membrane to release all the contents in the cells out in the medium. Then, once all the cell's components are released, you have all different organelles in that particular liquid. You're going to put it in a centrifuge tube. Basically, you put it in a tube and you start rotating it at a very high speed. And we call it centrifuge. Once you rotate it, those that are heavy, they're going to sink. My bad. Those that are heavy, they're going to sink to the bottom. And those that are light, they're going to stay afloat. So those that are light will be separated and stay at the top part. And the heavy cell components are going to stay at the bottom. So now you have successfully separated different parts of the cell. And now you can study each of these structure and component individually. Next, we know that cell can be divided into generally two types. We have the prokaryotes, which means no nucleus. And you also have eukaryotes, which means cells that has a nucleus and other membrane-bound organelle. Okay, and prokaryotes include the domain of bacteria and archaea, my bad, archaea. And we're going to focus on the structure of bacteria because that's usually what will be in the test. So bacteria is a prokaryote, so we're going to draw a simple cell like this, which is made of cell membrane. Right, membrane. And the purpose of the membrane is to enclose cytoplasm. Right? So cytoplasm is basically the region that's enclosed by the membrane. Cytoplasm. Sometimes you see the word cytosol. Cytosol focus more on the interior cellular environment. Which is enclosed by the membrane. Okay. Occasionally, this bacteria has an additional layer of protection. That's not a very good way of drawing it. And we call this additional layer cell wall. Okay, and this cell wall is generally made of peptidoglycan. Okay, and it is a rigid structure meant for protection and stability of the shape of the bacteria. For protection and maintain structure. Okay. Occasionally, around the cells, it has this tiny hair-like structure. And this hair-like structure are called fimbriae. Okay, F-R-I-M-B-R-I-A-E. So they are for short attachment structure that helps maintain the attachment of the bacteria at certain surface. Short attachment structure. And key. So we have the cell wall, and around the cell wall, occasionally you will also have a capsule. We call it a Glycocalyx capsule. And this capsule is made of polysaccharide or protein to help attachment to the surface. Right? So capsule, attachment. It can also be meant for protection. Right at the center, right, in the region of cytoplasm, we have nucleoid, which is basically the main chromosomes of the bacterial DNA. Nucleoid, it is the region where the cell DNA is located. And they will also have what we call an accessory DNA. You already seen it before. It's round, a lot smaller. We call this accessory DNA plasmid. Okay, so plasmid is a circular accessory DNA that codes for the gene that's not necessarily for the survival of the bacteria. It's not the main function of it. That's why we call it accessory. And we also have ribosomes in the cell, right? Ribosome. The function of a ribosome is to synthesize protein. Okay, and that's basically the general structure of a prokaryote or bacteria. And please remember the function of this following structure: cytoplasm, nucleoid, fimbriae, ribosome, cell wall, plasmid, capsule, membrane. Okay. And we also, okay, good. So now we can focus on eukaryotes. Right, Eu rhymes with new, so it has a nucleus. Eukaryotes can be divided into plant cell or animal cell. So these are the two main cell types that we need to focus on. Of course, these and protists are also eukaryotes, but the cell shape and structures are not the main focus. They share similar property, but we're going to focus on the similarities and differences of plant and animal cell. And we're going to start with an animal cell. And here, animal cell, usually irregular in shape, right? The outer layer is a cell membrane. Okay, another word for cell membrane, plasma membrane. We're going to go into detail of the structure of the plasma membrane later. So the function of it is to enclose the cell, right? Or to separate the cells from the external environment, or to create an internal environment for the cell. create an internal environment. Okay, and inside it we have the nucleus. Nucleus is itself a membrane-bound organelle, so this is what we call the nuclear membrane. Right? So nuclear membrane is a membrane, double layer, and around on the surface of the nuclear membrane you can see a lot of pores that allows material to go in and out of the membrane, and we call these pores nuclear pore. Right? So nuclear pore allows the transport of material in and out of a nucleus. Okay. Right at the center you see very dense DNA, and we call this nucleolus. Right? It is the location to synthesize ribosome or RNA, right? R stands for ribosomal RNA. And then around it you have this disorganized, loosely packed DNA around it, and we call this DNA chromatin. Right? Chromatins, by definition, they are non-condensed DNA and protein. Together we have nuclear envelope, nucleolus, chromatin, and nuclear pore. Together it forms the core of nucleus, which contain the gene necessarily for the activity of the cell. Okay. And as an extension to this nuclear membrane, we have the envelope system, and we have the RER and smooth ER. The differences between the RER and smooth ER is that RER has ribosomes on this membranous surface, and smooth ER doesn't have it. Right? So here, rough endoplasmic reticulum, right? The function of rough endoplasmic reticulum is to synthesize protein, usually secretory protein, and it can also be used to synthesize membrane phospholipid. Sometimes we will also say instead of synthesize protein, because we know that that's kind of like the ribosomes function, not necessarily the rough endoplasmic reticulum, but the ribosomes will attach to it and put the protein that are being synthesized into the rough ER for the rough ER to transport. So sometimes we will also say the rough ER's function is to transport the synthesized protein. Okay. So here, like I said, the function of this ribosomes is to synthesize protein. Ribosomes are made of rRNA and protein component. They are involved in the synthesis of protein through the process of translation. Okay. Here we have smooth ER. Smooth ER is the ER. And by the way, the ER itself is made of membrane, so please be aware of that. They are membrane. So here we have smooth ER. Interesting. Where's my green pen? Okay, use this one. Okay, smooth ER. Functions of smooth ER is quite diverse. They are meant to synthesize lipid. That's why organs that are used to make fat has a lot of smooth ER inside the cells. They are also important to metabolize carbohydrates, be it catabolically or anabolically. They're also important in the detoxification of drugs or toxins. And they are also important as a storage of calcium ion, which is important in the roles of contracting muscle. Okay. So please remember the function of smooth ER and be aware that they are involved in a lot of the metabolic pathway, such as synthesis of lipid, metabolism of carbohydrates, and the breakdown of drugs and toxin. Okay. So once the stuffs are being made, be it protein or lipid, and here we're gonna say the thing is protein, it will butt off from the end point of the membrane of the endoplasmic reticulum, and is going to form a structure called vesicle. Right, it's gonna butt off, and the things in there is something like a protein, right? So this is a vesicle. A vesicle is a membrane-bound sac. Think of it like a membrane ball. Okay? And these vesicles can be used to transport the made product. Okay? And it can be used to send to Golgi apparatus, which is also another membrane-bound organelle. Color looks kind of similar, so I'm going to change this to dark blue. Okay. So the vesicle is going to carry the protein, and then it's going to fuse with Golgi. Right. So vesicle is going to move, carry the material, fuse with Golgi, and the material is going to be transported into the Golgi. The function of the Golgi is to modify the material that it just received. So Golgi apparatus, sometimes the book will use Golgi body. They mean the same thing. Function is to modify, store, then transfer the protein in a new package, right? Which is the product of ER. Okay. Lysosomes, right? Sometimes the material that is making is a lysozyme. So if the lysozyme is being modified and is being released again in the vesicle, we call this thing a lysosome. So lysosome is a vesicle that carries lysozyme. Lysosome, right? So it contains the enzyme lysozyme. Okay, and this lysozyme is hydrolytic. It means it can be used to break down through the process of hydrolysis. Break down what then? Break down macromolecule. Okay. Some animal cells has a membrane sac, which is used for storage, and we call this a vacuole. Okay. A vacuole, if it's a food vacuole, that will be used to store food. Some protists, like for example Paramecium, they have a contractile vacuole, spelled it wrongly, contractile vacuole, right? Contract, it means that this particular vacuole can use contractions to pump water in and out of the cell in order to maintain water pressure. You don't want water pressure in the cells to be too high or too low as compared to the environment. to maintain water pressure. Okay, a few more things after the Golgi, the vacuole, the ribosomes, and now we need the powerhouse of the cell, right? Remember that in AP biology, please refrain from using analogy like powerhouse, battery, or things like that, because it's not scientific enough. So we need to be best specific. The question asks about the functions of the mitochondria, right? It behaves like a powerhouse, but please don't say that it is a powerhouse of the cell. Okay, minus space. See, where can I write it? Oh, I know. I'm gonna move this here. Mitochondria, move it to the bottom over here. Move vesicle slightly to the left. And there you go. I found space for mitochondria. And I'm going to use red. Okay, use some red color over here to represent mitochondria. So mitochondria, singular mitochondrion, but I'm kind of used to call it mitochondria. Mitochondrion, right? It is a double membrane bound organelle where cellular respiration happens. And it will generate most of the ATP for eukaryotic cells. Okay. Basically, this is the functions of the structure of a typical animal cell. Okay. Then we move on to a plant cell. Before I move on, remember that the structure of a animal cell, we had the plasma membrane, which is to create an internal environment. Of course, it's made of phospholipid bilayer. And we have the rough endoplasmic reticulum, which is to transport or synthesize protein. Function of the ribosomes, vacuole, vesicle, mitochondrion, lysosome, Golgi apparatus, smooth ER, and the structure of a nucleus. We have the nuclear membrane, nuclear pore, nucleolus, and chromatin. Okay. Now moving on to plant cell. Plant, we're going to change our tone a little bit different to a green color. The majority part of the cells remains the same. It still has a plasma membrane, and the function is the same. Right? So separate what's inside and outside of the cell to control and regulate what goes in and out of the cell. Phospholipid bilayer. It regulates what goes in and out of the cell. Right? Surrounding the plasma membrane right at the outside is cellulose, cell wall. Right? Cell wall, which is made of cellulose. It's a very tough and rigid structure, and notice that cellulose is outside of the plasma membrane. So if the plant cell is making cellulose and secreted or transported out of the cell, it needs to go through plasma membrane. Okay, so the function of a cell wall, just like the cell wall in the bacteria, it is to maintain the rigidity of the cell. Okay, so this cell also has a nucleus with a nuclear membrane, nucleolus, chromatin, and nuclear pore at the surface of the nuclear membrane. So I'm going to say this is the nucleus. Okay. Around it, you still have the SER and rough ER. So smooth ER, and then rough ER with the ribosomes around it. right? RER with ribosome, and smooth ER. The function is about the same. One thing that students always make mistake is that they think that a plant cell does not have mitochondria, but no, a plant cell also has mitochondria. Okay. A misconception is that they always think that a plant cell does not do cellular respiration, a plant cell does photosynthesis, and animal cell does cellular respiration. No, both animal cell and plant cell do have mitochondria in order to carry out cellular respiration. Because plant cell also needs cellular respiration to make ATP for whatever cellular activity that they need to conduct, right? For ATP. But the difference between a plant cell and an animal cell is that a plant cell is obtain the energy different from an animal cell, right? I forgot to even write the word plant cell over here. So plant cell, right? The way how they obtain the energy is that all plant cells are autotroph. All right, specifically photoautotroph. Right, using light is directed from the sunlight to convert that light energy into carbon source. And that organelle is chloroplast. So we're going to draw a chloroplast here. I'm going to use dark green. Right. And we'll go into detail of the structure of a chloroplast later. Chloroplast has triple membrane, right? And it contains the pigment important for photosynthesis. So it has it is a photosynthetic organelle, and it can convert light energy to chemical energy stored in sugar molecule, glucose or starch, depends on your perspective. And then on the membrane, one thing that you can take note of is that they occasionally has tubes on the surface of the membrane, and we call it plasmodemata. They are basically the tunnel in between plant cells to transfer molecules in between plant cells. So there are cytoplasmic channel, which means tube. It can pass through cell wall for connection to adjacent plant cell. Okay, and then we also don't forget that it has cytoskeleton. Animal cells also has cytoskeleton. Okay. And we will go into detail of cytoskeleton later. And the purpose of the cytoskeleton, one thing is to maintain the shape of the cell. And last is that all plant cells, in order to maintain the turgidity, has a large central vacuole. Right? So the purpose of the central vacuole is to store water. Okay. Breaks down waste material, and it hydrolyzes macromolecule. Okay. So to repeat, we have the plant cell. Okay. We have something that's unique to plant cell wall, large central vacuole, plasmodemata, chloroplast, yes. Okay, and something that's quite common between animal cells and plant cells, mitochondria. SER, RAV-ER, ribosomes, cytoskeleton, plasma membrane. Okay. Here I will make a note, only in plant cell. Those that are highlighted in yellow. Okay. Here basically we finished talking about the basics idea of cells, and moving on we will focus on plasma membrane and calculating cell surface area, as well as the endosymbiont theory. Okay, next time.